So moving on, next to is Streptococcus. It is also a gram-positive bacteria, but there is a slight change in the structure of Staphylococcus and Streptococcus. As we have already discussed that Staphylococcus is arranged in the grape-like structure or in clusters, but Streptococcus will be arranged in the form of chain. So the morphology or the study of shape or structure of Streptococcus is that it is a gram-positive bacteria, spherical and ovoid in shape. Ovoid, we can say that it is more or less of a egg-shaped structure, arranged in chains, no spore and flagella. In very young cultures, the capsule are noticeable. Now the meaning of we are seeing one word, that is flagella. So the meaning of flagella is it is a thread-like structure that allows to swim. A common example I can give of thread-like structure is spermatozoa. It has a thread-like structure which allows it to swim in the female reproductive organ. So it is a thread-like structure that allows to swim. But spermatozoa is not a bacteria. I am only saying about the shape and its structure. So flagella is a structure or a thread-like structure that allows to swim. So the culture and typical biochemical reaction, it is a facultative anaerobe. Already discussed about facultative anaerobe. That means it is an organism which can survive with or without oxygen. And when it will survive without oxygen, it will be via fermentation. Much higher nutritional requirement grow well on enriched media. We have seen in Staphylococcus that it grows in minimal nutritional requirement, but in Streptococcus the nutritional requirement for the growth of bacteria will be high and it grows well on enriched media. Now we are coming in contact with a word called enriched media. So the meaning of enriched media is contains nutrients that is required to support the growth of wide variety of organisms. That means enriched media is one that contains high amount of nutrients that is required to or that supports the growth of wide variety of organisms. And we can see that it is that the growth of Streptococcus can only occur when there is higher nutritional requirement. So enriched media contains a high nutritional amount which supports the growth of wide variety of organisms. Colonies 1 to 2 mm in diameter. In Staphylococcus it was 2 to 3 mm of diameter, but in Streptococcus it is 1 to 2 mm in diameter with a smooth, shiny, opaque surface. Different hemolytic zone around the colonies. Classification of Streptococci according to hemolytic activity. So hemolytic activity, that means refers to hemolysis, that means destruction of red blood cells that leads to the release of hemoglobin from RBCs into the blood plasma. So according to hemolytic activity, it is divided into three categories, that is alpha hemolytic streptococci, beta hemolytic streptococci and the gamma hemolytic streptococci. So discussing about the alpha hemolytic streptococcus, that is incomplete hemolysis, green zone around the colonies. So the hemolysis in alpha hemolytic streptococci will be incomplete, that means the destruction of RBCs will be incomplete. So the colonies will form incomplete and the color of the colonies will be like green and green zone will be formed in culture media. Moving on to beta hemolytic or pyogenic streptococcus, complete hemolysis, clear zone around colonies. That means there will be complete breakdown of our red blood cells. So there we can see clear zone around the colonies. So the colonies form in the culture plate or the culture medium, that will be clear zone. Next is gamma streptococcus, that is no hemolysation, no pathogenicity. So no destruction of red blood cells will occur in gamma streptococcus. So rating in the severity of major to minor. So the most destructive or most serious kind of hemolysis that occurs will be in beta hemolytic streptococcus, then will be alpha, and last one will be, that is the minor, that is gamma, where no hemolysation will be occurred. According to antigenic structure, group specific cell wall antigen, on the basis of antigen, streptococci can be classified into A to H and K to U groups. Chemical property, carbohydrate. The serological specificity of this antigen is determined by an amino sugar and protein. The foundation of subtypes within group. Major virulence factor of group A, streptococci pyogenes. Group A will be more than 100 serotypes, group B 4 serotypes, and group C 30 serotypes.